Thank you, and thank you all for being here on a Tuesday evening when you could be watching the State of the Union.
This will be better.
We'll see. So I've been at the Academy about a year and a half. I'm a coral biologist by training, and specifically my research focuses on global climate change impacts on coral reefs. So largely what I try to do, kind of the overarching question to everything that I do with my research, is addressing how coral reefs are going to be impacted by global climate change, and whether there are things that we can do to try to mitigate some of those potential impacts. So how these systems are changing overall. I have a background in coral biology and ecology, biogeochemistry, coral reproduction. Largely those are my areas of expertise. So tonight I wasn't going to focus too much on coral biology 101 or coral reef biology 101. Happy to get into that if we need to take a step back, but I was just going to jump into coral reproduction, assuming that we all kind of knew why coral reefs were important, and what a coral was. But again, at the end, if we want to go over any of those questions, happy to do that as well. So starting out with why coral reproduction is of interest to me in particular, I started studying coral reproduction about 12 years ago in my studies. And what triggered me to become particularly interested in reproduction was how quickly we were losing corals at that time. So when I was in grad school for my PhD, there were lots of scientific papers coming out saying, look, we're seeing unprecedented losses of corals because of bleaching, because of local eutrophication, pollution, ocean acidification, all these different things. It was as in 2003, which was 2 years before I started my PhD, there was a big paper that came out that we lost about 50% of the Caribbean over the last 30 years. And in 2011, which was the year I finished my PhD, there was a paper that came out on the Great Barrier Reef that said we lost 50% of that over the last 30 years. So in both ocean basins, huge losses in coral decline. And so I started to become interested in ways to restore what we were losing. And the primary way to restore what we're losing in any population is through reproduction. So you're trying to replace what you're losing at the same rate or at a greater rate so that you can even build up those populations even more. So one of the main focal points of my research over the last 12 years has been trying to understand what barriers there are to successful reproduction in terms of temperature impacts, ocean acidification impacts, pollution impacts, and whether there are ways that we can mitigate those and try to help shepherd corals through successive reproductive cycles. So getting into how corals reproduce in general, they're fascinating little creatures, although they look rather simplistic. They reproduce both asexually and sexually. So they're clonal organisms. So one coral consists of hundreds, if not thousands, of tiny little polyps that all reproduced from one another. Sorry, let me just... set this till later. Okay, sorry about that. So what started initially as one tiny little polyp, so you had a larva that preceded this, that metamorphosed and turned into one single polyp. And then that polyp budded and cloned asexually another polyp, which then cloned another one, et cetera, until you wind up getting a huge coral colony that's made up of hundreds to thousands of these things that are all genetically exactly the same. So that's one method of asexual reproduction, just within one colony. Another method, which takes advantage of the same technique, is called fragmenting, where they're kind of like regenerating starfish, where if you break off part of this colony, it will continue to grow into another colony. And corals utilize this strategy naturally. So if you have a hurricane that might come through and you have a branching colony and part of it gets broken off, it might tumble downstream, re-solidify to the reef, and continue to grow. And as long as that doesn't happen at catastrophic levels, that can actually be helpful to help corals propagate in different areas. So those are the two main modes of asexual fragmentation. And then they also reproduce sexually. And there's a couple of different ways that they do this. There's coral spawning and coral brooding. Coral spawning is you've probably seen it on National Geographic or other nature channels. It's very iconic. You usually see it happens once a year. It's a mass spawning event where these corals will release gamete bundles into the water column synchronously. So all up and down the Great Barrier Reef, which is 2,300 kilometers long, one night in late summer, all the corals release spawn into the water column. It's incredibly beautiful. It's amazing to see. And then those bundles will then have eggs and sperm in them. They'll break apart, they're fertilized, and then complete the life cycle. And we'll go into this a little bit more on the next slide. So that's spawning. And then there's also brooding, where instead of external fertilization, they're fertilized inside the coral colony and will develop into larvae inside of the actual colony, and then the colony will release competent larvae that are able to settle. So those are the two primary modes. They do all sorts of funky other things that people just notice anecdotally, and we don't know how often they happen in nature. But if we're just doing, generally speaking, those are the two primary sexual modes of reproduction. About 75% of coral species are spawners. So hermaphroditic spawners, meaning they release both eggs and sperm into the water column. What else do I want to touch on here? So one thing that we can talk a little bit about later when we talk about restoration, which we'll touch on, is that coral reef restoration techniques to date have largely taken advantage of this strategy here. So we're very good at this point. In the marine environment, restoration's been around 25, 30 years, primarily in the Caribbean. It's starting to gain traction in the Indo-Pacific. And it's based off of silviculture techniques and forestry that have been done for hundreds of years, where you take clippings of different genotypes that you might selectively breed and cultivate, and then you propagate those preferentially. So we can do that with corals. We have what are called coral gardens or coral nurseries, where you basically just take a colony, snip it up into a bunch of individuals, and then grow those into more corals and plant them back out onto the reef. So the majority of restoration uses this, and we're really pretty good at growing corals right now using this technique. One of the drawbacks to this is that you're propagating one genotype. And in a time where we're dealing with climate change and changing environmental conditions, that genetic diversity is so important to build up resilience in the population to be able to deal with stressors as they come in. So that's why there are a couple of groups, and the Academy is partnered with one that does sexual restoration work. And I'll talk about that again in a little bit. So most of the work that I focus on deals with sexual reproduction. So again, we have about 75% of coral species that are broadcast hermaphroditic spawners. So they, once a year, usually in late summer, it's linked to the lunar cycle. So it's usually a certain number of days after the full moon. In August, or if you're in the austral summer, then usually November, December. And different species go different days after the full moon. Some will be 5 days after the full moon, some will be 7 days after the full moon. But generally once a year, they will release what we call gamete bundles. So they're hermaphroditic, so those bundles have both eggs and sperm in them. And what's kind of cool, evolutionarily about them is that they have to be packaged together if you are in an aquatic environment, because eggs by nature are very, very buoyant because they have so much fat and so many lipids in them. Sperm are not. And so if they released eggs separately from sperm, the eggs would shoot to the surface and the sperm wouldn't, or they'd have to swim up there and expend a lot of energy, and they might never meet up. And so they're packaged together. There's a sperm bundle in the middle of the, or a sperm package in the middle of the bundle, and then it's lined with eggs outside, and then the eggs basically carry the sperm packet to the surface. And then about, depends on the species, but anywhere from 30 to 60 minutes after release, that'll dissolve and break apart, and the eggs and the sperm are released at the surface, and they can all mix together and fertilize one another. So that's what's supposed to happen. So gamete bundles float to surface, bundles break apart, fertilization occurs. So then you have a developing embryo that, again, depending on the species, days to weeks, it will develop into a larva, a planula larva. And I have some pictures. So this is what a larva looks like. These are developing embryos. So this is just in a 2-cell stage. This is an egg that hasn't been fertilized. These are those gamete bundles being released. They're usually pink or orange in coloration. And then this, which we haven't gotten to yet, is it's a larva that has metamorphosed into a primary polyp that will then start to bud asexually into a coral. So what's cool about these is, so they spend days to weeks in the water column. They're completely covered with cilia. So that's how they swim around. And largely, their trajectory is determined by ocean currents. But they do have the ability to swim up and down in the water column to influence their trajectory. So once they are ready to settle, they'll swim down to the ocean floor and start probing around for a good place to set up shop. So another thing that I find super cool about these, even though these are about the size of a sesame seed, and they're very visually unstimulating, like they don't actually look very cool. There are some larvae, like fish larvae, sea urchin larvae, things like that are gorgeous to look at. These are not super interesting to look at. However, they're fascinating because they have an oral and an aboral end at this stage. And when this thing actually metamorphoses into this, what will happen, its aboral end is full of sensory mechanisms, like photoreceptors, chemical receptors. If you think about a coral, that basically once it makes a decision to cement itself to a reef and start calcifying, that thing is then stuck there for probably hundreds of years. So that's a very deterministic, important decision of where it's going to settle. So they're actually extremely sophisticated in terms of sensory mechanisms at this aboral end, so that they can probe the environment and figure out where is a good place to settle. And some of the things we think cue them into good places are light. So we know they have photoreceptors. They don't have eyes, obviously, but they have opsins. They have different genes and proteins that do sense light. They'll gravitate towards or away from light, different colors. They'll actually gravitate towards things that are red. This type of algae, this pink that's coating this rock here, is called crustose coralline algae. They really like it to settle on. A lot of marine invertebrates do. And there's some indication that part of its color, part of it is bacteria that live on the surface of the algae. Part of it is chemicals that the algae release that the larvae then sense with their chemical receptors. So anyways, point being, these things are chock full. And if you watch them when they're trying to settle under a microscope, because they're coated with cilia, they swim, the cilia beat and they kind of turn like this in the water column. They swim like little torpedoes. And what they'll do is they will swim with their aboral end first because they're sensing things, and they'll go down and they'll probe the environment and just kind of touch the bottom and like explore. And if they don't like that area, then they'll shoot back up and they'll swim around, and then they'll sense out another place. And then and they just keep doing that until they find some place that indicates to them that this is a good place to start to metamorphose, lay down a basal plate and start calcifying and cement yourself to the reef for the next decades to centuries. So that is probably a little too much information about that, but I think it's pretty cool. So that's this process here, settlement. And then the last part of the life cycle that we still consider an early life history stage is this post-settlement growth, because they're just one polyp here. And this is actually, there's a series of bottlenecks here that kind of winnow down the population. But this is a really important one because there's a lot of mortality post-settlement of, if you think about everything that's on a reef, if you've seen pictures of the reef floor, there's so much growing on that reef. There's turf algae, macroalgae, soft coral, sponges, cyanobacteria. I mean, you would never find an inch of reef that wasn't covered by something. So the name of the game is, as soon as you get there, you need to grow as fast as you can and claim as much territory as you can. And the better you do that, the better your chances are of survival. And just like in a rainforest, there are different strategies to do that. So some will just try to shoot out sideways and just claim lateral area. Some species will shoot up and try to shade things, like some trees do, to try to kill whatever underneath, and then they can grow laterally. There's different strategies by different species, but this is another critical stage to survivorship. And then if they survive around a couple of years, they're considered a juvenile, and then they'll grow back up into an adult that then hopefully completes a spawning cycle and contributes to the population. So that, in a nutshell, is the coral life cycle for a spawner. And our best guess, I mean, you can imagine during these stages how much predation and how much loss there is, and then additional loss here. So our best guess of how many individuals actually make it through this is 1 in a million. And so anything that we can do as scientists to help up those odds, particularly right now when corals are really challenged, is of benefit to coral populations. So that's one of the main focus areas of our work with SECOR, which is the sexual restoration organization. So before I move on, just since we're here, a couple of things in terms of challenges right now to this being a successful process. What we think is happening is that, well, obviously this is becoming much less successful. And so part of what we think is happening, which happened a while ago in the Caribbean, and we'll see if it plays out in the Indo-Pacific too, is that what's supposed to happen is this synchronous spawning, where probably through pheromones, corals are sensing each other in terms of when, on that night following the full moon in late summer, at a specific time of night, and some populations have been tagged down to the minute, like 7:13 p.m., all of those colonies will release. Because you can imagine if one colony would go and another one would go an hour later, doesn't do anybody any good. These things don't self-fertilize. They have to fertilize different genotypes. So if one goes on one night, one goes another night, doesn't do anybody any good. So it has to be synchronous in order for this to work. And so because populations of corals are becoming very depauperate, you don't have those dense thickets and pheromone or chemical communication, or however they're kind of syncing themselves up, happening the way that it should. And so there's been a breakdown in the Caribbean in synchronicity that has been a huge hurdle for successful sexual reproduction of corals. So there's only a couple of species that actually still do synchronous spawning in the Caribbean. And so that's another thing that we try to do with SECOR is we go out, and again, we can't really do anything if one spawns one night and one goes the next night, but what we can do is we spread ourselves out very, very far, even on different reefs. And we all monitor, we send teams to all sorts of different reefs, all monitoring stuff. And if we get spawn, we all bring it back to the lab. And then we can get 100% fertilization in the lab. We know how to do that now, what kind of conditions they like. We can rear them to larvae and get 100% survival here. We can even get 100% survivorship or settlement because we know what they like. So what we do is we take that crustose coralline algae, that pink algae, and grind it up, or you can extract chemicals, compounds from it. If you grind it up, people call it fairy dust, and then you sprinkle it on whatever you want them to settle on, and they'll settle. And usually you can get really, really high rates of settlement. And so what we're doing is trying to jumpstart them through all these things that might not happen in nature, get them at least to this point, and then rear them out so that hopefully they're big enough that when we put them back out on the reef, they have a fighting chance. So those are some of the things that we're trying to do to intervene. I think that's all I wanted to mention there. So, again, I apologize for my voice. I was out with pneumonia last week, and I'm just still a little off color. So, what we've done to help part of Hope for Reefs, a large part of Hope for Reefs, is our partnership with this organization, SECOR. And SECOR, a lot of people think it's spelled with an A, S-E-A. It's not. What this stands for is Sexual Coral Reproduction. And it is the only, I think, restoration organization that focuses on sexual reproduction as opposed to fragmentation in coral nurseries and coral gardens and things like that. And so what we do with them is we go out into the field. We know when different species are supposed to spawn, and we go and we monitor them. And so this is a Cropera palmata, which is elkhorn coral from the Caribbean. It's endangered now. These are pictures of me from my PhD work in the Florida Keys. But what we do is when we know they're going to spawn, we'll net them. And then they release these positively buoyant gamete bundles up into that float up. This is all spawn that's going up into this little collector cup. Every single one of those tiny dots is a gamete bundle that has probably thousands of sperm, millions of sperm, and hundreds of eggs. And then we then take this from different genotypes back into the lab, separate the eggs and sperm, and can cross-fertilize them under whatever conditions we want and rear them out. So this is generally what we do with SECOR. We have permanent sites in 3 places in the Caribbean, so Curacao, the U.S. Virgin Islands, and Mexico, and they are trying to expand internationally to Guam and hopefully elsewhere in the Indo-Pacific. Sorry, not internationally, that's all these other countries are international too. I meant to the Indo-Pacific and Guam. And then hopefully elsewhere in the Indo-Pacific. And we work on a bunch of different species. So different species go at different times of year. So I was involved last year with a brain coral in Curacao in June. That was the only species that went. It's called Diploria labyrinthiformis. It's a brain coral that goes specifically in June. Then we go back in September, and there's about 10 or 12 species that go in September. So different times at different locations at different times of year, targeting different species, but it's the same concept for every species, every location. These are some pictures from Curacao last year. So one of the really neat things about this partnership with SECOR is that we go and do this restoration work, but we also partner and train stakeholders from a variety of different countries. And so what you see here is a mixture of SECOR staff and Cal Academy staff. So me, Val, a few other people are actually SECOR staff. And then the rest of these individuals are people from various Caribbean countries. We had representatives in June from the Dominican Republic, Puerto Rico, U.S. Virgin Islands, Mexico, all over the place in the Caribbean that had nurseries or coral gardens in place already, And we wanted to teach them how you add the sexual component in to complement the asexual component. So they're there learning all of these techniques so that they can take it home and add it into their already existing restoration programs. So, okay, one thing that we haven't talked about is what happens when you settle them onto a substrate and then you put them back out onto the reef. And so one of the biggest hurdles with any restoration program, and one of the largest criticisms of most restoration programs, is we know how to do things really well in a lab in a controlled environment, and then you take those things and you put them back out into a super degraded environment, which is why they weren't doing well in the first place, and why would you ever expect them to do well now? So obviously a big part of, in order for restoration to be successful, is acknowledging that you have to mitigate those sources of environmental stress that caused the decline in the first place. Easier said than done, particularly in some countries. But one of the criticisms with restoration across the board is that a lot of times post-transplant survivorship can be sometimes quite low. And that's because you're putting them back into a degraded environment. So one of the ways to try to get around that is to try to put out as many as possible. And so we're trying to figure out, and another hurdle to getting more out onto the reef is that it's very labor and time intensive. So you imagine you're settling all these things onto little tiles, and historically, so this little pyramid thing is something new that Sea Corps and Cal Academy came up with. But for the last 25 years, we settled them onto little terracotta tiles or concrete tiles that were two-dimensional. And then you would go out onto the reef and you would have to cement that tile to the reef or epoxy it to the reef. And it's very labor and time intensive. You have a team of divers out there literally mixing epoxy underwater and trying to fit these things to the reef. And so that's the bottleneck to scaling up, because right now with most restoration efforts, we're very good at restoring things on a hectare scale, but ecological destruction is happening at kilometers, at thousands of kilometers. And so we just can't keep pace with what's happening. And so what Cal Academy and Sea Corps are particularly interested in tackling is this question of how do we scale this? How do we bypass those hurdles of time and labor impediments to make this ecologically meaningful of a practice? And so one of the ways that we're starting to try to tackle this problem is through substrate design. And so instead of using two-dimensional tiles that you have to fix to the reef, what they've done is designed these, they look like tetrapods. We call them tetrapods. So they're 4 prongs, 3 in the bottom, 1 sticking up from the top. The reason that they're grooved is, so these have been conditioned in the ocean, so that's why they have this algae on them that corals love, that pink stuff. And they've been conditioned in the ocean for 1 to 2 months. And then we bring them in, and they're grooved because corals love little, coral larvae love little nooks and crannies to settle in. They like when it's rough. So they'll probably settle in those nooks and crannies. And the idea with these substrates is that instead of going out and having to cement them, you could scatter them like farmers kind of scatter seeds in a field. And these things have been shown to move. So they'll tumble a little bit with current, but they've been shown to move less than around 50 centimeters. I believe it's around less than 50 centimeters in 6 months, which isn't very far. And so as the corals start to grow, so this is a picture, you can't see it very well, but this is one arm, this is an arm, and this is an arm. So this is one tetrapod here. And this is a coral that's growing onto that tetrapod. So it started, it grew out, and it'll start to grow onto the reef substrate. So the idea is that with these prongs, these things might be able to wedge themselves into spaces, and we could go out and instead of having to physically cement them, we can just kind of, even from a boat, not even from diving, you could just throw them overboard and then cover a lot more spatial scale and hopefully make this, scale this up to ecologically meaningful levels. So that's one way we're addressing the scale issue. These are just some more pictures from Curacao last year. Again, tenting the coral releasing spawn, and then fertilizing things in the lab. And incidentally, one thing that's cool about this species, this is the brain coral, Diploria labyrinthiformis. So with different species, you'll know that they're going to spawn different ways. Some species do what we call setting, where usually there's one bundle per polyp, and the bundle's kind of down in the stomach of the polyp, and right before it's going to release, it'll come up and it'll just sit in the mouth. And so you'll know that a colony is going to spawn if it's set, which basically just means you look really closely and you can see in the mouths of the polyps these little pink bundles in different patches of the colony. This coral doesn't do that. It will set for like a couple of minutes, not very long. Some species will set for half an hour, and it gives you time to know they're going to spawn, go grab a net, set everything up. This one doesn't do that. But what's cool about this species is that the butterfly fish know they sense something about this species that indicates them, because as soon as everything spawns, it is a feeding frenzy on the reef in terms of predation activity. We used to wear hoods because you would get worms and all sorts of stuff in your ears. So what we'll do when we're watching to see if the species is spawning is follow the butterfly fish. And they will literally go up to a colony and sniff it. And you'll just see them like in pairs all over sniffing the colony, and then they'll swoop off to the next one. And if you see them stay at a colony and then a bunch more come, you know that one's going to spawn, and then you'll go over and you'll scare them away and you'll net it. And you better hope you didn't get a butterfly fish in your net, because it would have eaten everything. So that's it about the field work that we're doing. And what I'm also trying to do here at Cal Academy in conjunction with the Steinhardt Lab is build a lab where we can spawn corals in captivity. And so this has only been done at one other place in the world, at the Horniman Museum in London. They've been successful at doing this 4 years in a row. Corals are notoriously difficult to keep in captivity, let alone spawn in captivity. And part of the reason is that we don't fully understand the suite of environmental factors that is important for gametogenesis to happen. We know that spermatogenesis probably only takes a couple of months, 1, 2, 3 months maybe. Oogenesis, to produce those eggs, that's a huge energy investment on behalf of the coral, and in some species that'll last up to 9 months. And so you have to keep the corals happy in terms of nutrition, water temperature, water chemistry, flow, lunar cycle, lighting, incident light levels, all these things year round in order to try to trigger one spawning event. So what you have to do, generally what we think cues corals to the day that they will spawn, is what gets it to the month is changes in seasonal temperature throughout the year. And so they tend to spawn in late summer when the temperature warms up enough that it will probably trigger the maturation of eggs like a month before. And then in that late summer when that temperature peaks, that's the month they're going to spawn in. So that's one thing we have to replicate in the lab, are these seasonal oscillations in temperature between summer and winter. Once you get it to the month, what will get it to the day is that lunar cycle. So we have to now simulate full moons and new moons and changes in blue light intensity and red light intensity. And then what will get it to the hour is incident light level. So now we're mimicking sunrise, sunset, and absolute irradiance. So those are the primary, and then we have to make sure that they're nutritionally replete throughout the year, and the water chemistry is good, and all these other things. So it's a tricky business, but I feel like we can do it. So if they did it in London, we can do it here. So we are working, so this person, Jamie Craggs, was the individual who was responsible for having this succeed in London, and we contracted him. He's been advising us in the process so far. And what we're building is a pilot system to try this out. And then if we get it to work, the hope is that we'll get maybe some donors interested enough in this that we could then scale and build a much bigger system. Because wouldn't it be cool to have systems staggered so that you could get coral spawning every couple of months? And so if we just shift the system in a way that they respond, then we can probably get them spawning several times a year, if not every month. So this is where the system is so far. And this has been a labor of love, trust me. But one of the, I feel like this is being weird. Is this okay? Anyways, so the space that I was allocated is in the aquarium holding room, which I'm sure you're all familiar with. So in the back of house, you go up those little stairs, and there's that or it's not the quarantine room, it's the holding room. And there used to be those two large blue tanks with corals in them. So what we've done is kept one of those tanks for asexual reproduction, and we've taken out the second tank. And what we're building there is basically the equivalent of a photography dark room. So it's a room within a room, and the logic or the reason behind that is light pollution. Because we have to work within the confines of another space, we don't have complete autonomy over the lighting. And light pollution, this person in London found out, is one of the biggest reasons why they'll spawn asynchronously. So we need to have complete control over that lighting to make sure things are going as synchronously as possible. So what you're seeing here is if you were standing in the holding room and you had one of those big blue tanks at your back, what you're facing is what used to be the space for the other big blue tank. And there's 3 walls that have been built. The 4th one isn't in place yet because we're still working on it. But there's black walls here, one large one at the back and one at the side. And then there'll be one put here, and it'll just look like a big black box. And then just like a dark room, there'll be another door. So you have to open, close, and be completely dark before you actually enter to really try to control that light pollution. And then the main parameters we're manipulating in here are temperature, again for those seasonal cycles, and lighting, because we're mimicking lunar phases and incident light level at different times of day. And so this is kind of the status. This picture was just taken a couple of days ago, so this is the status of the system right now. We have tanks in place. Water's been cycling for several weeks. We'll probably put test corals in within the next couple of weeks so that it starts cycling with some biological material in it. Lights are all in place. And we have to go, here's just another picture, collect corals to put into the tanks, which we're doing at the end of February. And so for that first spawn, what we want to make sure we do is get corals that are gravid, that we know are going to reproduce, because there are a lot of duds, unfortunately, out on the reef, and we don't want that. So what you can do about a month or 2 months before you know corals are going to spawn, if you break a branch off, you can actually see the eggs fully developed in there as little pink dots. And that's the way you know that coral's gravid and it's reproductively viable. So we have a collections trip at the end of February. So in Palau, which is where we're going, corals, the spawning species that we want, we're going to target two staghorn corals that are spawners, a Acropora tenuis and a Acropora hyacinthus. And those typically spawn in April. And the full moons this year are March 31st and April 30th, and we want to collect our corals about a month before that. So we're going around February 28th, beginning of March, to collect corals to bring back, with the hope that if we keep them on that cycle that they were supposed to be on in Palau, on March 31st, or around then that they should spawn with the native corals back in the field. And we have collaborators that will be in Palau monitoring whether or not those spawn so that we can see how synced up we are. We may also collect a couple of brooding species just so we have 2 spawners and 2 brooders in the tank, and we have a couple of different things to work with from a research standpoint. So that, I think that, yeah, that's my last slide actually. So that's the status of where we are with that right now. And there's a lot of different applications we can do with this. So part of what we can do is test concepts for all the sea core and field restoration work that we do. So if we come up with, if this works here, and we're able to expand and build a couple of systems that are staggered, then we have not one opportunity a year in the field where we can test a bunch of ideas, but we could have opportunities every month where we can test different substrate designs. We can test ways to maximize growth, ways to maximize settlement, things that we could do and then feed back into all those field practices to try to maximize survivorship in the field. So that's one restoration application in a way that we could partner these lab and field efforts. Another application is just basic fundamental biological, like evolutionary and developmental science. There are a lot of organisms that have model systems for them, like Drosophila and zebrafish, different things like that, that we know so much from a molecular and genetic standpoint about how these systems work and function. We know next to nothing about corals because they're notoriously difficult to keep and to complete the life cycle in nature. We don't, or in a lab, and we don't have a model system for corals. So if we're able to complete the life cycle, we might be able to understand molecular mechanisms underpinning calcification or underpinning bleaching responses and things like that, so that we could then figure out if there are ways that we could mitigate them, if there are medical applications, all sorts of different fascinating questions we could use that for. And there was one other I was going to mention. It escaped me, but it'll come back. But all sorts of different things we can do with this. So I'm really excited about it. We're really close to the pilot system being up and running. Again, we're kind of limited by space in Steinhardt. And so what we're hoping is that this will be a successful endeavor, that then we could maybe get some funding to expand and build another facility at some point. So I will leave it there. I feel like I chattered for more than a half an hour, but we have plenty of time for questions or conversation, whatever you'd like. I'm okay, thank you.
Right. So I'm sort of thinking about the California condor here, and of course it's sort of different, but the condors got to be rare, and they brought them into captivity, and then they figured out how to breed them, and then they took that part of nature.
Right.
And nature is still screwed up because of all the lead that's out there. How are you going to deal with the challenges of the environment? I mean, let's say you can reproduce them, but how are you going to get them to like the crabby conditions after they've lived in the lab?
Yeah, I mean, that's the challenge, right? And so there's a couple of different ways that people are trying to address that. One, and actually this is kind of splitting the community, the scientific community. It's a contentious issue right now because there's people who think we should do things like genetic engineering of corals to produce stress-tolerant corals, bleaching-resistant corals, selective breeding of corals, and then outplant those. And there are others who vehemently oppose to that and want to focus on mitigating the root source of the problem. So I think it's your fundamental conservation discussion that's being had in the coral community right now. But there are people who are actively researching and breeding super corals, which you can do a couple of different ways. One is just selectively breed. So there's a lot of genotypic variation in terms of response to these stressors. And so just doing experiments to figure out which ones are more tolerant than others, and then selectively breeding those and using those in your restoration efforts. That's one strategy that's been used in silviculture for a very long time. And then you can also actively manipulate them. So corals, as you might know, are an animal that has an algae, a symbiotic algae that lives in its tissue, and then also coated by bacteria. Corals are long-lived organisms, so it's difficult for the actual animal to evolve generationally in time to keep up with this, but you can, in a lab, evolve the symbiodinium, or the algae that live inside, or the bacterial communities that have much shorter generation times to withstand stress, and then inoculate a coral with those. So they have successfully evolved heat-tolerant algae in the lab and inoculated corals with them, and I believe we're in the stage of testing whether or not that actually increases the thermal tolerance of the coral. So those are some things that are being looked at. And then the obvious one is just sounding the alarm about about mitigating these stressors to begin with. The biggest one right now, the elephant in the room for coral reefs everywhere, is temperature, global warming, and climate change. And so promoting and trying to get people to understand that no matter what we do from a restoration standpoint, or even an artificial or genetic engineering standpoint, that these are band-aids that buy us time, but we need to fix the source of the problem. And that's going to be garnering political will to shift towards sustainable sources of energy. But yeah.
And that's what we're learning right now in the sustainability class, the ways that each of us can impact that.
Yeah. And I do remember what I was going to say before, which kind of links into your question, or which is another application of this, is that another thing that is gaining a lot of traction is cryopreservation of coral material. So there's a department at the Smithsonian that has developed since 2005 cryopreservation techniques for corals. Right now they're very successful at cryopreserving sperm, and they're working on developing techniques to cryopreserve eggs and larvae, and then whole corals. So once you start zapping eggs, which are very lipid heavy with liquid nitrogen, odd things happen that shouldn't. So those techniques require a little bit of methods development, but they're very good at cryopreserving sperm. And so what they've been doing, I think they've banked 16 species of the estimated 800 coral species. So what is that, 5%? Ish, something like that. And so if we're able to breed things in captivity, and then we're entrained in those practices, one thing that we could potentially do here is serve as another bank. So that germplasm has been distributed around the world to different repositories, and we could potentially serve as one of those where we're breeding things and then banking that. And so then if we need in a certain area in the world to add genetic diversity of a certain species, just like they do with seed banks, you could pull out some sperm of something and add it into the mix and create genetic diversity. Because that's another way we fight this, how to deal with environmental degradation. The more diversity you get out there, the better your chances are. Yeah?
I'm just curious, besides the heat, how much is the acidification because of the CO2 dissolving in the ocean that's causing?
I love that you asked that, because that's actually my other area of expertise, is ocean acidification impacts on corals. So ocean acidification, we think, has probably decreased coral calcification somewhere in the realm of 10-15% over the last 100 years. So I did an experiment a couple years ago that was published in Nature last year that showed that ocean acidification has already impaired calcification capacity on a reef. And it also interferes with reproduction. And one of the ways it interferes with reproduction is by forcing things to become hypoxic or hyperoxic, and particularly sperm. So it slows sperm down, it slows sperm motility, and then fertilization doesn't happen as easily. So it's definitely impairing things already. It hasn't been as well studied as temperature because it's not as visible and it's not acute. So I often liken them to like a heart attack versus cancer, because a temperature has this capacity to, within the span of a couple of weeks, because it's so acute, devastate thousands and thousands of kilometers of a reef. Ocean acidification, to what we know today, it doesn't cause mortality. What it does is hinder growth and hinder reproduction. So it hinders the ability of those populations to come back once they are decimated. So bleaching will wipe them out, temperature will wipe them out, and then they can't come back as quickly because they can't reproduce and grow. So they're acting on two different time scales. One, OA is more chronic, and temperature is much more acute. But we know it's already impairing them. It's just, in terms of statistics, we don't have as good of a finger on it. But the pH of the ocean's increased about 30% over the acidity of the ocean has increased 30% over the last 100 years, and that's had a huge impact on calcification potential. Yeah, it's decreased by 0.1 pH units, which from 8.2 to 8.1, which doesn't sound like a lot, but because it's a logarithmic scale, that is 30%. So.
I was wondering, how do corals themselves avoid self-fertilization in the wild, since all the polyps are releasing their bundles at once? And then how do you avoid that when you bring back those bundles to the lab?
That's a good question. Mechanistically, I don't know that I know how they avoid it. They just don't. So very, very few species will be able to self-fertilize. The ones where we do see self-fertilization, it's usually at a rate of around 2 to 3%. But in terms of, usually between the egg and the sperm, there's binding proteins on the surface of an egg that the sperm will recognize, and then there's a reaction between, like an acrosomal reaction between those. So I don't know if it has to do with recognition of those proteins. But yeah, mechanistically, I don't know why, but it's not something that we have to actively prevent. It just doesn't happen. So that's actually one way when we're doing experiments that we test to make sure we didn't have any contamination is we'll put a control that is a self-control. And if we see any fertilization in there, we know that we messed something up, because you should get 0 to 1% fertilization at most. I don't think that fully answers your question, sorry, but.
In this tank that you're building down in, basically, you're controlling temperature, you're controlling light. What are you feeding the coral?
That's a question for Rich Ross, our aquarium biologist. I believe it's a mixture. So they'll eat, corals are also heterotrophic, so they feed autotrophically because the algae that live inside of them photosynthesize, but they are heterotrophic as well. Generally what they eat is somewhat contingent on the polyp size, which changes from species to species. So they will eat things as large as worms, like marine worms. They'll eat things as large as rotifers, copepods, things like that. In past labs that I've dealt with, we a lot of times fed them Artemia, like sea monkeys. You can feed them oyster eggs, which are really lipid rich and very nutritionally replete for them. Mixtures, usually it's a mixture of plankton and zooplankton that you'll feed them. Here I don't know specifically what that mixture is, but we can ask our aquarium biologist. But usually it's a mixture of plankton and zooplankton, and the size of them depends on what species you're feeding.
Who else?
Are you going to discuss coral brooders?
Sure. Yeah, I'm happy to. Today?
I just don't know what it is. I know spawning, fragmentation. What's a brooder?
So touched on that a little bit at the beginning, but basically what a brooder is something that is a species that undergoes internal fertilization. So in all the spawners, the eggs and sperm are released into the water column, and there's external fertilization. In a brooder, if they're dioecious or gonochoric, which just means separate sexes, female and male, The male will release sperm, and then the sperm swim over to the female. The female takes the sperm in, and there's internal fertilization. And then the larvae actually develop inside of the parent colony. And then when that colony broods, we don't call it spawn, we say when that colony broods, it actually releases larvae that sometimes just kind of crawl out of the polyp mouth over the surface. They're not really super buoyant, and so they'll just kind of release slow and crawl out over the surface of the colony. And most times, because those larvae are released fully competent, so if you're a spawner, you're releasing things that then have to develop in the water column for days and weeks, and so they'll be carried by oceanographic currents, you know, hundreds sometimes, unless they're caught in an eddy, sometimes they go hundreds of miles away. For a brooder, they're releasing larvae that are ready to settle right away. So a lot of times you'll get settlement right next to the parent colony. So you'll sometimes see these gregarious or like clusters of brooders because they're all kind of reproducing together in one area. So brooding is, we think, not as prevalent as spawning. So we think 75% of coral species are spawners, fewer brooders. But for some reason, there's this hypothesis right now that the brooders are more resilient and robust to environmental stress because they have more control over that reproductive cycle. And so in the Caribbean, over the last 30 years, we've seen a huge shift. There's a lot of factors playing into why this happened, but loss of spawners and increased dominance of brooders. And so it's kind of this joke in the coral world that these brooders are going to take over the world. But a lot of times brooders tend to be smaller, more isolated species that might not build up reef framework as much. So it's important to have those spawners there too, because the spawners tend to be those branching species that give all that like arborescent complexity that the fish and everything like to hide in. And a lot of brooders are kind of smaller that don't really give as much topographic complexity.
So as I understand it, the corals that we have in our Philippine coral reef just survive on photosynthesis, and we don't feed them. I remember asking Charles that question once when I was at the reef. So if he's going to feed planktonic food to your system, how are you going to prevent that from being filtered out? Because I know that the Philippine coral reef has a filtration system that would remove.
So our system is separate from the Philippines coral reef system, and a lot of times what they'll do if it's a recirculating, I haven't talked about this with Rich yet, but in past systems what they'll do is you'll turn it off. You'll turn off that circulation, feed them, leave it for an hour or two, and then turn it back on and let it cycle. So there are like delineated times during the day where you actively feed them. I'm sure you're right about the PCR exhibit. Again, that's a question for Bart or Rich. I'm not actually sure if we feed in there or not, but if you heard we didn't, you're probably right. Yeah.
And the other question I have is the corals that we have growing, they don't spawn? Is that because we don't have the right light?
No, so what's really, one of the reasons why I think this is not undoable or unfeasible is that anecdotally, so the corals we have in the PCR exhibit, the brooders brood all the time. So there are Pastelopora damicornis, this guy and this guy are in the PCR exhibit. You'll see little spat or little baby corals all around this all the time. So these are very active brooders in our PCR exhibit. We have some of these too. And anecdotally, some of the biologists have said they have seen some of the corals release spawn every now and then, but no one really was invested enough at the time to note when or what species or which colony or anything like that. But I have heard from talking with some of the biologists that they've seen it happen. Okay. So it's very encouraging that, because we use especially because we use artificial seawater here, right. that the lighting and the temperature and the chemistry might be right for gametogenesis.
So from Palau to San Francisco is a long way. What do you do to the corals to ensure that they're going to be intact when they get back to San Francisco for transport?
So we're talking about that right now, and we're consulting with the guy from London. So he transported his from Singapore to London. And what you do is you keep them in styrofoam coolers, and you actually suspend them, one coral per thing, from the top, so that it's suspended in water and can kind of move around and jostle, and it's not hitting the sides, so to prevent it from too much mechanical damage. And then we're going to put temperature loggers in there so that we know what happens to the temperature throughout the time that they're shipped. So what Jamie, the guy from London, said happened to his corals, and this is part of why you want to do it a month before, is that those corals are going to be in darkness for probably 36, maybe 48 hours. And what will happen is they'll start to dark bleach, and the temperature will probably drop. So they'll start to get stressed, they'll start to dark bleach a little bit. So Jamie mentioned that he would see some paling. And so if they're losing some of that algae, which is their food source, so when you bring them back in, you have to transition them pretty carefully into really good water quality. And then if corals have gametes and they're stressed, a lot of times what they'll do is resorb their gametes as an energy source. And so we have to prevent that from happening. So what he's recommended that we do is if we see the bleaching or the paling happen, as soon as they get back, we just give them oyster egg. We give them as much food as we can so that they feed heterotrophically and they don't resorb their gametes. And then hopefully we can mitigate that and they'll come back up and be okay. So.
And are they going to be shipped on commercial airlines or?
Yeah, we check them.
Oh my God.
It's pretty heavily permitted and regulated. We have to meet with a lot of people along the way. Yeah. You can check them as baggage, done that before, or you can, a lot of airlines will ship things as well. So if you're flying United, you can go to United Cargo, and you can either check it or go to United Cargo and just ship it, but they'll send it on the same flight so that it's all, because from a permitting standpoint, you have to accompany it. So it has to go with us as we go.
Wow.
So, yeah.
More questions?
Why is the Caribbean so far advanced in this process over the Indo-Pacific?
And then from a restoration standpoint?
No, from a pollution standpoint.
Oh. So we had a huge coral die-off in the early 80s, 82, 83, and part of it was because of warming associated with El Nino. And then, so there was a big outbreak of a disease that caused die-off of diadema, which is an urchin that's very, very common in the Caribbean, that grazes down algae, most of the algae. And so there was this huge diadema die-off in the early 80s that they think triggered all this algal growth that kind of overtook corals. So when the corals were compromised by that warming event, and there weren't any herbivores there to keep the algae at bay, the algae basically just took over, and we saw this phase shift happen from coral-dominated to algal-dominated that has never really restored. So there's different schools of thought as to why that originally happened. Some think that it was the disease that broke out in the diadema. Some think it was just eutrophication because it's a smaller ocean basin, so more pollution, more direct anthropogenic impacts, overfishing. But the removal of herbivores earlier on was a big one.
Hi, I could ask you 1,000 questions, but just one that relates to us as docents. So we have the opportunity to speak to all sorts of people every day when we're volunteering here. What's a good takeaway message that we can, in a short span of time, share with people that we come across in the aquarium who might be curious about some of the extra things that the Academy is doing? I
think that specifically related to reefs, I think that what's really neat about what the Academy is doing right now, a couple of different things, but with Hope for Reefs, and to my knowledge, we are one of the few organizations in the world that still gets funding for basic exploration from a marine standpoint. So in an academic setting, that would never happen. And so for the mesophotic and the twilight reef stuff, it's really fascinating, I think, that we have this commitment to exploring these systems, like the depths of the ocean, that we know nothing about. And I think we're in a unique position as an institution to be able to do that basic exploration. That will then set the foundation and the groundwork for academic and other institutions to ask targeted, hypothesis-driven research questions, which is what they'll get funded to do. So I think that's really an important part of what we do as an institution. And the other is trying to tackle these. Again, we're in a unique position to have the flexibility to be able to tackle some of these high risk but high reward questions. Again, you can't do that in an academic setting. If you submit an NSF grant to try to tackle a high risk but high reward question, you would never get funded. And so we have this privilege to be able to ask questions like, how do we scale restoration? Great, we can restore a reef the size of this room. We just lost a half of the Great Barrier Reef. Like, what do we do with that? So I think understanding the unique position the Academy is in terms of trying to ask really questions that really would move the field forward is really special. That's what struck me since I've been here over the last year. Because I've worked in government, I worked in academic, and I worked in other nonprofits before. And since I've been here, that's one of the most refreshing things about being here. We don't really have any bounds to the science we can do. Like, they're letting me build a coral spawning lab. If that's not high risk, high reward. So.
Other questions? Just to piggyback on what Rebecca said, I want to just read, how many of you have started the sustainability training process? Okay, about a third of the room. You need to know that our focus is changing as docents, as volunteers, from being people who discuss what we already know to empowering people to make an impact through regular, everyday actions that will, if we all work on it together, make an impact and inspire them to do that. So in a different way, we too can message. While Rebecca's down doing her incredibly important research, and this academy has funded all of this. Well, actually, I'm sure she's got funding from many different sources, but she's getting to do this here. We can tell this story and others every time we're on the floor and talk about the way we as individuals can make a positive impact on this. So I highly encourage those of you who haven't done sustainability to sign up for the summer class when it starts.
And it just, I mean, we hit on this twice already, but just for a third time, it really is, as a scientist, a very unique opportunity, because almost everywhere that I've been so far in an academic setting, you'd say, well, I want to do this. And they'd say, well, you're never going to get funded to do that. You can't do that. What if you do this? And you were always brought back down. And then in the government, I worked for the Australian government for 4 years, and it was the same thing. You'd say, well, I want to do this. And they'd say, oh, okay, like everybody would love to do that, but we can't do that. And so in here, it's just, if you say, well, what if we did this? And you get this overwhelming sense of support, as long as it's not irresponsible. We don't do irresponsible science, but if it's something that could move the needle, there's a huge amount of support behind you for, okay, well, why don't you try to do that? And then you could do this after. And so that kind of limitless support for what you could do is very unique to this institution.
Rebecca, thank you so very much.
Thank you.